Good afternoon friends and welcome to the CEC EduSet lecture. Today we are starting a series on the molecular biology, and for that we have a guest, Dr. Charu Dogra Rawat, who has been teaching molecular biology for the past 12 years in the college in the Delhi University College, and we will be exploring with her the physicochemical basis of heredity. We will be looking at the observations and experiments that have led to the discovery of DNA. We will also be looking at how DNA became that chemical entity which is responsible for carrying the blueprint of an organism. So I welcome Dr. Charu Dogra to continue with her lecture.
Thank you so much. So I begin the series with this molecular biology lecture in which I talk about the structure and function of a very important molecule, which is DNA. And so at the end of this lecture, the learning objectives, which is that you will be able to identify and present the experimental evidences that DNA conveys genetic information. You should be able to talk about the discovery of the structure of DNA. You should be able to tell the physical and chemical features of the DNA, and you should also be able to tell about the types of DNA. I began my lecture by quoting a very famous molecular biologist, Francis Crick, of Watson and Crick fame, who said that almost all aspects of life are engineered at the molecular level, and without understanding molecules, we can only have a very sketchy understanding of life itself. So basically, to know about life, we need to study molecular biology. So what is molecular biology? Molecular biology is a branch of science concerning biological activity at the molecular level. That is, it involves the study of interactions between the molecular components that carry out the various biological processes in the living cells. So basically, it's an interplay of the biomolecules residing in the cell that renders it the biological activity. There are four major classes of biomolecules: proteins, which form the largest part of the biomolecules inside a cell. It renders the enzymatic activity to the cell and also are the structural component of the cell. Carbohydrates, the source of energy, they also are structurally important. Lipids, they are the energy storage molecules, and they are the important constituents of the biological membranes. And nucleic acids such as RNA or DNA that possess the genetic information that can be inherited or passed on from one generation to another. Earlier, the search for the genetic material started when the scientists started taking heredity in a biological sense. So they were looking for basically some substance that must be responsible for the passage of traits from parents to the offspring. They also understood that for a substance to do this, it must be stable enough to store information for long periods. That is, because it has to be inherited from one generation to another, it must hold the information and it must also transmit this information. It must be able to replicate accurately. That is, it must make copies of itself which can be passed on from the next to the next generation. And it must be capable of change to allow evolution. Now you understand that variations that are inside the DNA, that only leads to the larger process, which is basically the evolution. In the early 1900s, when William Fleming discovered chromatin, which was later called as chromosomes, he discovered that the chromosomes are basically responsible for the inheritance. Subsequent studies related to the chemical structure of chromosomes identified that they contained DNA, but they also contained proteins. Now, before we decipher that out of the DNA and the proteins, what was responsible for the genetic information to be passed on from one generation to another, we would just look at like where did this DNA come from? How was it isolated, or when was it isolated the first time? In 1859, Friedrich Miescher, he basically identified DNA, although he didn't know about it. He collected pus from the bandages at a local hospital, the pus that contained white blood cells. He treated them with a weak alkaline solutions and isolated a material from the nucleus, and then that he named as nuclein. This nuclein is the crude form of DNA, but he didn't know about it. He could also identify though that it was made up of hydrogen, oxygen, nitrogen and phosphorus, and there was a unique ratio of phosphorus to nitrogen. In 1874, separated the nuclein into the protein and an acid molecule. So Friedrich Miescher was the first person to identify the nuclein or the DNA. It was followed in 1909, and almost Phoebus Levine, he worked for 3 decades in finding out the chemical composition of this nuclein material. And he identified the components of DNA, which was phosphate, deoxyribose sugar and a nitrogenous base. He also showed that the components were linked together in the order phosphate, sugar, base to form units. He called each of these units a nucleotide. The DNA molecule consisted of a string of nucleotide units linked through the phosphate groups, which are the backbone of the molecule. The possible bases which were there, they were divided into 2 categories, the purines and the pyrimidines. The purines could be adenine and guanine, and the pyrimidines could be cytosine, thymine and uracil. The nucleoside is the ribose sugar or connected with the bases, and then it adds on the phosphate group to form a nucleotide. So chemical composition of this nuclein material or DNA was basically identified by Phoebus Levine. He also proposed a tetranucleotide hypothesis that suggested that the 4 components of DNA occur in approximately equal ratios. So he talked about that the nucleotides, which are different because of the addition of the bases, which is A, G, C or T, the entire DNA contained equal ratio, which is A is equal to G is equal to T is equal to C. The 4 components of the DNA were present in equal ratios. And he suggested that the DNA was made up of this repeating tetramer. So they understood that the proportion of all these 4 bases, the nucleotides, are equal in the DNA, and this tetramer just repeats itself. So the DNA is basically AGCT, AGCT, AGCT. So the tetramer keeps on repeating itself, and that's what the DNA is composed of. So by far, the DNA contained millions of nucleotides arranged in a very monotonous and a predictable fashion that could have no meaningful information content. So there was no meaning at all. All it contains was a repeating tetramer of 4 nucleotides, which is A, T, G and C. However, if you look at the proteins, they are made up of amino acids. The 20 amino acids at that time, probably 16 amino acids were discovered. But the proteins are made up of amino acids and linked together in diverse sequences. So that promised to carry a genetic code. And therefore, the scientists diverted their studies towards the protein molecule to be identified as the genetic carrier. The structure of DNA was too simple and too regular to contribute to any kind of variations that is occurring. Therefore, the attention focused on the protein as a probably hereditary structure. The idea that DNA may be the genetic material came from 2 important experiments. One was in 1928, the Frederick Griffith's transformation experiment, followed by in 1944, when Avery, MacLeod and Carty pursued these transformation experiments to find out that what exactly is the genetic material. So we'll have a look at both these experiments and discuss them. In 1928, the Frederick Griffith was studying on the Streptococcus pneumonia, which causes the pneumonial disease, and he found that there existed 2 strains of this particular organism. One was the rough strain, which was basically uncapsulated. It did not have any covering outside, and therefore it was having a rough appearance, but it was non-pathogenic. It was avirulent in nature. The actual strain which was causing the disease was the S strain, also termed as the smooth strain, because it secreted a polysaccharide capsule around it and therefore it gave a smooth appearance to the colonies when they were grown on the solid media. And they were the one which were disease causing or virulent or pathogenic bacteria. He was working on these two bacteria, and then what he found out was that he started off with the virulent S bacteria and he injected the mice and then observed what happened after a few days. When he injected this virulent bacteria, for obvious, the mice died after the few days of injection. And he could also isolate the living S strain cells which were found in the heart. In another experiment, he injected the mice with the non-virulent R strain of Streptococcus pneumonia. And again, as expected, the mouse that was injected with the non-virulent, non-pathogenic R strain survived and it lived. And there was no bacterial cells which could be found in the heart of the organism when it was opened. In another experiment, they heat killed the S strain. So, heat killing the virulent S strain, basically when injected into the mice lived. And there were again no bacterial cells found in the heart, which clearly showed that if you apply heat to a virulent bacteria, then it is becoming a non-pathogenic or its activity is lost. So, heat is killing the pathogenic activity of the S strain. In another experiment, what he did was he mixed the two strains. He mixed the R strain and the dead S strain bacteria and then injected it into the mice, and that was the most surprising result. The result was that the bacteria died, the mouse died, and the living S strain cells could be found in the heart. So that was very surprising result, because the virulent strain, which is the S strain, is dead, and the non-virulent strain, which is non-pathogenic, is living. But when both of them are mixed together, they are causing the disease and the mouse dies, and you are getting a living S strain cells which is found inside the heart. So based upon these experiments, the Griffith concluded there was something from the dead S cells that transformed the R cells into more virulent S form. So although the S strain cells were dead, they still contained some molecule or something which was transforming the non-virulent, non-pathogenic R strain to become disease causing. The process he termed the transformation, and the unknown substance was termed as the transforming principle. Avery, MacLeod and Carty in 1944, when came to know about this experiment, they experimented and isolated different compositions, but they worked on the cells and they did not work on the mice. So they took the lysate from the bacteria and they divided it into different components, and each component they tried on the cells. So they removed the lipids and carbohydrates from a solution of heat killed cells. Proteins, RNA and DNA remained. So they took the S strain cells, live cells, they lysed them and they just removed the lipids and carbohydrates components of the cell. So what was remaining was protein, RNA and DNA. And then they did a sequential elimination. First they treated it, they subjected the solution to the treatments of enzymes to destroy either the proteins, RNA or DNA. So they set up 3 experiments. In one of the experiments, the sample contained everything but the protein. In the second experiment, the sample contained everything but the RNA. And in the third experiment, the sample contained everything except the DNA. So the enzyme proteinases, ribonucleases and deoxyribonucleases in separate experiments removed the relevant component. Then they added a small proportion of each sample to a culture containing R cells, and they observed whether the transformation has occurred by testing the presence of the virulent S cells. So they already knew that the dead S cells get converted into the virulent cells by the addition of R. So which component is actually causing it, they checked it. The sample in which there was no protein and the samples in which there was no RNA, the transformation occurs. That means that in both the flask, in both the cultures, the live S cells could appear. So there was live S cells in both the things. but where there was no DNA or there was an absence of DNA, no transformation could occur or no S cells could appear. That was a very, very direct evidence that actually DNA is causing this transformation to occur from the dead S cells or heat killed S cells to become the live S cells to be there. Then what happened, they also identified that which part of the DNA, they isolated, they purified the DNA and showed directly that it is responsible for causing this. So what they did was they isolated the cells and then they isolated the DNA from it. They lysed the cells, they isolated the DNA, and the DNA probably contained a cap gene which was responsible for causing the capsulation, which was absent in the R strain but present in the S strain. So chemically they can find out that the presence of this cap gene can induce the formation of the capsule. So they isolated the DNA, the sheared DNA, and they added the DNA to the R cells. So when this cap R genes got inside the R cells, they recombined with the cap R which was present and which was not responsive, which was basically not causing the capsulation to occur. When the recombination occurred and cell division occurred, then basically the R cells could form the capsule. So that was a very, very direct evidence that the DNA, the purified DNA can cause the transformation, or it can basically cause the R strain to transform into S strain which is there. Now Avery and MacLeod, the conclusion was basically that the transforming principle was indeed DNA. The scientist called Erwin Chargaff was very, very fascinated by the discovery of Avery and his group. In fact, he quoted that I saw before me in dark the contours, the beginning of a grammar of biology. Avery gave us the first text of a new language, or rather, he showed us where to look for it. I resolved to search for this text. So he was very fascinated by Avery MacLeod discovery that the transforming principle was DNA, and he worked towards to find out that what is the composition of this DNA which is responsible for this transformation to happen. He pioneered the paper chromatography of nucleic acids and determined how much of each of the component nucleotides was contained in a DNA sample. So as the table shows that in different organisms, if you look at the proportion of A, G, C and T, and if you find the base ratios, that A to the ratio of T and G to the ratio of C is always 1. So if you remember that the Phoebus Levine, when he proposed the tetranucleotide hypothesis, he said that the DNA is a repeating tetramer. So the A, G, C and T are probably in equal ratios, and they are occurring, after every, the tetrameric is the one unit and it is occurring again and again. But now when Erwin Chargaff worked on it, on the paper chromatography of nucleic acids, he found out that the ratios are not equal. Each species differed in the amount of A, C, G and T, but within the species the proportion of each are identical, no matter what tissue of the DNA it is extracted from. The Chargaff's rule basically states that A is not equal to T, is not equal to G, is not equal to C. However, A is equal to T and G is equal to C. The A plus G, or the ratio of the purines to the ratio of C plus T, or A plus G divided by C plus T, which is purines ratio to the pyrimidines, is always 1. Or in other words, the ratios of A is to G and T is to C were always similar to each other, whether they were greater than or less than 1. So that was a very, very important discovery as far as the structure of the, or the components of the, or the distribution of the bases or nucleotides inside a DNA is concerned. Because the A and T and G and C, the ratio of purines and pyrimidines was found to be equal to 1, but not that all the 4 nucleotides are in equal proportion. Chargaff did find this, but he could not make sense of these, what he said as striking but perhaps meaningless irregularities. So about these findings, he could not interpret anything, or he could not conclude anything, and he just ignored them as perhaps meaningless regularities. Another important experiment was done by Hershey and Chase, which finally validated that DNA is the genetic material, because earlier the scientists were thinking whether the DNA or the protein is the genetic material. Avery's experiments, Avery and his group's experiments, quite confirmed that DNA is the genetic material. Hershey and Chase finally validated it by saying that protein is not the genetic material. They worked on an organism called as the bacteriophage T2. You can see the picture of a micrograph of a bacteriophage T2, and the structure is given on the right hand side. The DNA is present inside a capsule, a cap of proteins, which is formed of protein units. So there is a capsid, there is a sheath, there is a tail fiber. These are all protein components, and inside this head is present the DNA. So this is a molecule, a bacteriophage T2, and if you look at the lytic life cycle of a virulent phage, it follows of any other pathogenic lytic life cycle of a phage, the T2, which basically attaches to the E. coli and injects its chromosome. So the protein parts remain outside, only the DNA is injected to go inside the bacteria. The bacterial chromosomes break down the phage chromosome and then it replicates. The expression of phage genes present on the phage chromosomes produces phage structural components which dominate the cellular machinery of the bacterium. The progeny phage particles assemble and the bacterial wall lies and releasing the progeny phages. So that's a typical lytic life cycle of any virus, any phage such as T2. So it infects the bacteria, injects its DNA inside it, the protein coat remains outside, only the virus chromosome tends to replicate inside the cell, and then more and more progeny are formed and released by lysing the bacterial cell wall. The experiment done by Hershey and Chase was commonly called as the blender experiment, and what they did was they prepared the radioactively labeled T2 bacteriophage. So as we saw in the bacteriophage, there was a protein covering outside and there was a DNA which was present inside the head. Now chemical composition of protein and DNA differs because protein contains sulfur whereas DNA does not, and DNA contains phosphate whereas it does not contain sulfur. So they prepared this radioactively labeled bacteriophages by growing the bacteriophages in either P32 containing medium, which is a radioactive phosphorus, or S35 containing medium, which is radioactive sulfur. So if the bacteriophage is grown in a P32 containing medium, then only the P32, the DNA is labeled by this radioactivity. When the bacteriophage is grown inside the S35 containing medium, then the radioactivity is incorporated in the protein coat. So they can obtain the differentially radioactive labeled phage particles, one in which the phosphorus was labeled and one in which the sulfate was labeled. So basically one in which the DNA was tagged and another in which the protein was tagged. Subsequently, what they did was they infected the bacteria with the radioactively labeled bacteriophages, and then they just blended it in the normal kitchen blender. That's why it's called as the blender experiment. So they just blended it in a kitchen blender so that the bacteriophages which are attached to the bacteria are removed, and then they followed the radioactivity which is recovered in the host and passed on to the phage progeny. What they could found was that this S35 or the sulfur could not be found in the progeny. The protein that was labeled could not pass its content, could not be going on from the one generation to another. However, 1/3 of the progeny contained the radioactively labeled phosphate. So this was again a very clear evidence that at least protein is not the hereditary material and the DNA is passed on from one generation to the other. So they concluded that the protein is not the hereditary material. Following this, in 1952, looking at, this was now established that DNA is the genetic material. So all the scientists now started focusing on the elucidating the structure of the DNA, because DNA became the most important molecule in the cell, and they tried to find out the structure of the DNA. And in that series, the final nail in the structure came from the Rosalind, Franklin and Maurice Wilkins experiments in 1952. The technique which they used was termed as X-ray crystallography, And the Franklin could discover that DNA could crystallize into two different forms, an A form and a B form. We'll talk about more of these multiple conformations down in our lecture. She could obtain the crystals of the two forms separately, and they produce the interpretable X-ray diffraction patterns of them. So, X-ray diffraction technique basically involves the X-rays to be thrown onto the molecule, and then the deflected X-rays are taken up on a screen, and they give rise to the X-ray patterns such as given for the A form and the B form. It's called as the photo 51 because it got famous by that name, because it gave very clear thing about that how the DNA looks like, or what is the structure of the DNA. So, it's called as the photo 51. The interpretation from the Rosalind, Franklin and Maurice Wilkins work could be very straightforwardly that the structure is probably helical. It is composed of more than one polynucleotide chain. It could be either 2 or 3. The phosphate groups lie on the outside of the structural unit on a helix of a diameter about 20 angstrom, and approximately 3.4 angstrom thick purine and pyrimidine bases are regularly stacked next to each other perpendicular to the helical axis. If you see these X-ray graphs, they are giving a complete X kind of a X letter, which basically says that it is a helical form and it is a repeated form. And if you can see, there are two bands which are lying at a equidistant from each other, which is basically the stacking of the base pairs, which is shown here. So, their interpretation was basically which was very important for the structure of the DNA, which was given later by the Watson and Crick. But as I was telling you, now the DNA became the most important molecule of the cell, and it was shown to be carrying the genetic information in subsequent generations. So, all the research focused to find out the structure of the DNA. And in the 1953, in fact, Linus Pauling, who is famous for elucidating the structure of protein, also proposed a model of DNA. And it was published in the Nature, and what he proposed was that it was a triple helix model of DNA, that 3 strands are intertwined together, and this is how it looks like. So, he showed that the bases are towards outside and the phosphates are towards inside, and probably there are 3 strands which are which are intertwining each other in a triple helix model. But this model clearly had a lot of flaws and errors. The first one, that the phosphate groups were shown to be the neutral molecules. The phosphate groups are ionized, they get to ionized inside water, they cannot remain at the neutral molecule. So, if they are proposed to be present inside towards the inner side of the DNA double helix, then they are going to repel each other. So, in the proposed model as shown above, the phosphate groups were shown to be the neutral molecules. They are organized in the core for the helix, and the negative charges on the oxygen would repel. So, it is not a correct thing. The bases were facing outwards again, which was not possible because the bases there are 4 different types of bases, and that would give a very distorted structure for the DNA, because if outside there is a variation in the structure, then the entire DNA double helix should be distorted, and it will not give the X-ray diffraction pattern which was obtained by the Franklin and Maurice Wilkins, because it was very consistent. The bases, the diameter which was shown was very consistent, that it appears as a rod. So, if you have different bases facing outwards, then it is not possible, because that will give rise to a very distorted structure, because somewhere A will be outside, somewhere G will be, one is a double ringed structure, one is a single ringed structure. So, it was not actually very feasible, it did not look very promising. And then some of the van der Waals distances which they calculated appeared to be very small than what would be present inside the DNA. So, Linus Pauling's model was not accepted, and then came in April 1953, James Watson and Francis Crick, who proposed the molecular nature of nucleic acids. But if you look at it now, what is left to discover? They already knew that the hereditary molecule is DNA. It has been substantiated and validated that the molecule which is causing the inheritance is DNA. The DNA molecules are polynucleotides. It has become clear. The DNA has a double helical structure with alternating sugar phosphate molecules and the bases are in the middle. So, if it does not, the X-ray diffraction pattern clearly showed that the bases are supposed to be in the inside of the DNA double helix. And the double helical structure had a sugar phosphate backbone, the alternating sugar phosphate, which was already deciphered. What they don't know was basically that how the bases are arranged in the middle. So, the bases are inside, but how are they arranged? So, the approach which they took was to find stereochemically most favorable configuration compatible with the X-ray diffraction data produced by Wilkins and Franklin. The approach which they took was molecular modeling, and they modeled that what will be the best or stereochemically best, energetically favorable positions of the bases or the molecules which will give rise to the X-ray diffraction pattern of the Wilkins and Franklin, which will correspond to that data. They proposed this kind of a model. The model in which the two polynucleotide chains are wound each other in a double helix. So, the two chains are wound around each other in a double helical structure. The fundamentally building block of DNA is a nucleotide. The phosphate is joined to a sugar and to which a base is attached. So, if you look at this, this is how a deoxyribose sugar is attached to a phosphate group and is attached to a base. This is the nucleotide. If you see, the difference between the deoxyribose sugar and the ribose sugar is the position at 2' of the sugar molecule, where OH is replaced by H. OH is present in the ribose sugar, whereas it is replaced by H in the deoxyribose sugar, and the phosphate group is attached to one part of the sugar molecule. The sugar and base alone is called as the nucleoside. The bases, as already told, fall into two classes: the purines, which are derived from a double ringed structure, and pyrimidines, which are derived from the single ringed structure. The purines are adenine and guanine, and pyrimidines are cytosine and thymine. We are talking in DNA right now, so it's cytosine and thymine, which are derived from the single structure, and if you can see, then those rounded nitrogens are the point at which it attaches to the sugar molecules. So, they are arranged in such a manner, the nucleotides joined to each other through a 3' hydroxyl of the deoxyribose of one nucleotide and the phosphate attached to the 5' hydroxyl of another nucleotide. So, if you can see, the nucleotide one unit attached to another unit to attach to third unit is basically by this phosphate linkages, in which one 3' hydroxyl group of one sugar is attached to the 5' phosphate hydroxyl group of the another nucleotide, and this linkage is the phosphodiester linkage. This create, as you can see, is the sugar phosphate backbone of the polynucleotide chain. This kind of arrangement imparts inherent polarity to the DNA. So, what do you understand by the polarity? The polarity is that at both the ends there is an asymmetry. At one side there is a 3' hydroxyl group which is exposed, but on the other side there is a 5' phosphate group which is open. So, the polarity is defined by the asymmetry of the nucleotides and the way they are joined. The free 5' OH or 5' phosphate at one end and 3' phosphate or 3' OH at the other end. So, one polynucleotide chain is formed by the addition of one nucleotide to another nucleotide by phosphodiester linkages, leaving the two ends asymmetric, with at one end 5' hydroxyl group exposed and at the other end the 3' phosphate group or 3' hydroxyl group which is exposed. The bases, how do the bases pair? Now, the bases which are formed inside the double helix, how do they pair? The base pairing or the Watson-Crick pairing derives from the complementarity of both shape and hydrogen bonding properties between the A and T and the G and C. So, it was basically shown that A matches with the T and G matches with the C, and A and T makes 2 bonds within each other, we will talk about them, and G and C makes 3 bonds within one another, And these base pairing is derived by the complementarity of the structure, because as we thought that why did we discard the Linus Pauling model of DNA? The basic reason was that he showed the bases to be outside, and when the bases are outside, then because of their structural differences it will be a distorted structure. But since the DNA is not distorted and found as a linear cylinder kind of a molecule, so that means that the bases when present inside have to fit in such kind of an arrangement that from the outside it is kind of a very uniform structure, it is not very much distorted. The bases are in preferred tautomeric forms. The A and T matches to form the 2 hydrogen bonds between them, and G and C matches to form 3 hydrogen bonds between them. AT base pair and GC base pair have exactly the same geometry, giving the uniformity on the outside because they are located inside the double helical structure. The two-fold axis of symmetry that relates the sugars and all 4 base pairs can then be accommodated within the same arrangement without any distortion in the structure. So, this was the most suitable kind of an arrangement, which was also reflected in the X-ray diffraction pattern of the Wilkins and Franklin. So, the bases are towards inside, A matches with T and G matches with C, but still when they were trying to do it, there was some kind of distortion which was occurring, and so they came up with the next thing. So, now you can see the base pairing, that why adenine only pairs with thymine. The adenine, as you can see the structure, and the thymine, they have the potential of 2 bonds to be formed between them. One has a hydrogen, a proton, and another has a negative oxygen, and when they come close together they can form 2 bonds between them. And if you look at the base pair of guanine and cytosine, then guanine has potentially 3 molecules, 3 ionic forms which are outside, the O minus, the H plus and H plus, and in cytosine also it produces 3 ion which is H plus, N minus and O minus, and so if when they come together they have the possibility to form 3 bonds within themselves. But if you now have a look at the adenine and cytosine, this is the structure of the adenine and it was giving rise to a proton and an N minus, and this is cytosine. If you see, they tend to repel each other because the plus and the N minus are going to repel each other, and such a pair cannot be formed. Similarly, if you have a look at the guanine and thymine, the guanine and thymine have conflicting ions on the outside when they come close together, and therefore there will be a repulsion and no base pairing can occur between guanine and thymine. So, it was quite logical as far as the electronic composition is concerned, or as far as the energetically favorable stereochemical possible structures is concerned, that A will form a bond with T and G will form a bond with C. The A will form 2 bonds and G has the possibility of forming 3 bonds which is there. And these bases are then stacked one above the other to neatly on top of each other between the 2 helical sugar phosphate backbones. And this double helix is then stabilized by hydrogen bonding between the base pairs and electron cloud interactions between the bases in the helical stacks. So, there are 2 forms of energies which is basically keeping the DNA in its structural form. One is the hydrogen bonding which is occurring between the base pairs that are present inside the DNA double helix, and another which is giving the stability to this kind of a molecule is the stacking, which is the stacking of base pairs one over the other, which is keeping it intact, otherwise they will just collapse. So, these electron cloud interactions between the bases in the helical stacks is basically giving the structure of the DNA. So, the Watson and Crick model of DNA also proposed that the 2 chains are anti-parallel. Now, everything what they were looking at was okay, all the bases are inside, the sugar phosphate backbone is outside. The inside bases, they know that there is a possibility that A and T can combine together and G and C can combine together in 3 forms, but still they were not able to find, they were not able to get the structure which was reflected in the X-ray diffraction pattern. And a major observation which Watson made was that the 2 chains are running anti-parallelly in direction. So the sugar phosphate backbones are on the outside of the helix and the bases are on the inside, stacked perpendicularly to the long axis like the steps of a spiral structure. So both the strands, which are double helically intertwined over each other, are actually running in an anti-parallel direction. Now what do you mean by the anti-parallel direction? So in the anti-parallel direction basically means that there is one strand which is basically running from the 5' phosphate group to a 3' hydroxyl group, and on the other strand there is a 5' phosphate group facing the 3' hydroxyl group and 3' hydroxyl group facing the 5' phosphate group. So the two chains which are intertwined together are basically in the anti-parallel direction. So one is running from 5' to 3' and the another facing is running from the 3' to the 5' end, and in that manner all the bases which are present inside can be stacked one over the another irrespective of whether it is AT or GC, and they can give a very non-distorted structure or a very constant structure from the outside. So anti-parallel orientation is a stereochemical consequence of the way the bases pair with each other. So you can see that A:T and G:C pairing results in complementary relationship between sequence of bases on two intertwined chains. So there are two chains, one is running from the 5' end to 3' end, the another is running from a 3' end to the 5' end. In between you have bases which are lying inside. There is G basing pair with C, the T basing pair with A, the G and C forms 3 hydrogen bonds, T and A forms 2 hydrogen bonds, and irrespective of whether it is G or C or T and A, structurally when they are stacked over another they fits perfectly. So it's a very perfect double helical structure which would give rise to the double helical, the X-ray diffraction pattern which was obtained by the Franklin and Wilkins. So Watson and Crick model of DNA, the physical parameters were basically that there is a residue on each chain every 3.4 angstrom. The structure repeats after 10 residues, approximately 10 residues on each chain, that is after 34 angstrom. The distance of a phosphorus atom from the axis is 10 angstrom. So that means the width of the DNA strand is approximately 2 nanometers, and we will see down in another lecture that how this 2 nanometer condenses to form a higher order structure. So this is a very basic structure, very first level structure one can say of the DNA, that's how the DNA is the structure of DNA looks like, but that's not how it looks like in the cell. In the cell it has different levels of organizations. So we will talk about this in subsequent lectures in our series, that how this DNA is arranged. You know the cell division, that how it is arranged in the interphase nucleus and how it condenses to form the metaphase chromosomes which are present. So everything which is, all the chromosomes is composed of DNA along with the proteins, but the structure of the DNA condenses to form such kind of higher order structures as we say. So the structure of DNA proposed by Watson and Crick was the first order structure of the DNA, that's how the double helical structure of a DNA looks like. So DNA is usually a right-handed helix. It can be, when it's a helical structure, it can be a right-handed helical structure or a left-handed helical structure. But DNA usually is a double-handed it's usually a right-handed helical structure, and as you can see there is an existence of two grooves, the major groove and the minor groove. So alternating major and minor grooves occur because of this right-handed helicness in the DNA. The edges of each base pair are exposed in major and minor groove. So major and minor groove are the turns where the DNA bases are exposed towards the outside. They are present inside forming the bonds, but at these grooves they are exposed towards the outside, and they create a kind of a pattern of hydrogen bond donors and acceptors and van der Waals surfaces that identifies the base pair. So what is the significance of this major and minor groove which is occurring? So the significance of this major and minor groove, you know that the DNA interacts with a lot of proteins. the DNA in a cell has to perform function, the hereditary function, and again in the subsequent lecture we will talk about that how DNA passes on this information from one generation to another, which is basically intrinsic in its structure. How does it do that? So, to do that it has to associate with proteins. For the DNA to decode whatever is encoded inside its information in its sequence, the proteins has to act upon it and convert it into say RNA and then further to proteins. So, the proteins have to interact with DNA. Now, to interact with DNA, the proteins will have to unwind the DNA. And unwinding does not mean that they will have to open up the double helical structure and then only interact with the exact sequence of the DNA structure. Initially what they do is they interact with the bases which are exposed from the major and the minor grooves. So, this major and minor grooves and the bases which are exposed create a kind of a pattern of hydrogen bond donors and acceptors which are sensed by the proteins which need to interact with them. So, basically if you look at this, this is a picture showing when the G and C base pairing occurs, then on the major groove side there is an acceptor, there is a creation of an acceptor, there is a creation of a donor, and there is a, so in that pocket, in that major groove, there will be an environment where there will be two acceptors, one donor and one a proton. So, that kind of a thing, that is what is sensed by the proteins and probably the first interaction occurs. So, in the minor groove for example, when G and C corresponds, there is an acceptor, there is a donor and then there is an acceptor. So, in all the four combinations, the ions which are, or the ionic environment which it represents at these grooves are shown in this picture. That there is a, for example in C and G if they correspond, or for example in A and T if they correspond, then there is an acceptor, there is a donor, there is an acceptor, and there is a methyl group which is towards the major groove, and towards the minor groove there is an acceptor, there is a proton and an acceptor. So, the protein basically senses this environment. It does not know whether there is a G or a C or a T which is present. It does not know the sequence as such. It will know about the sequence when it will unwind the DNA and start interacting with the sequence by itself. But initial interaction is based upon this major groove, whether they interact from the major groove or whether they interact from the minor groove. The basis of interaction is basically this, that they allow the protein to unambiguously recognize the DNA sequences without having to open and disrupt the double helix. So, based upon the kind of an ionic environment of major and minor groove, the proteins can sense what the bases are present in those grooves, and before interacting directly with the nucleotides or directly with the bases or directly with reading the sequence of the DNA, they can interact through these major and minor grooves based upon this kind of an ionic environment. So, based upon this discovery of the structure of DNA, so we followed the history of discovery of structure of DNA. The three scientists, Francis Harry Compton Crick, James Dewey Watson and Maurice Hugh Frederick Wilkins, were given Nobel Prize in Physiology in Medicine in 1962 for their discoveries concerning the molecular structure of nucleic acids and its significance for information transfer in living material. Franklin, we talked about Franklin, Rosalind Franklin, and she worked with Frederick Wilkins to produce the X-ray diffraction data, which was very substantial for Watson and Crick to find out the structure of DNA. But she could not be given Nobel Prize because she died before this, and then they did not have the provision in any case to give Nobel Prize to 4 people at that time. But in any case, they also did not have provision to give the Nobel Prize posthumously. So, Rosalind Franklin also, although she contributed a lot to the discovery of the structure of DNA, she could not be given the Nobel Prize related to that. The last part of today's lecture basically deals with the multiple conformations in DNA. So, in a cell, in a structure, in a cell, there are different kind of conformations of DNA occur. The DNA double helical structure is the structure which is found inside the cell, inside the nucleus, but based upon usually the water content, there could be different conformations which it can take up, and this was initially seen by Franklin Wilkins, who could isolate the Franklin and Wilkins, who could isolate the different crystal forms of two kinds of DNA, which is the A form DNA and B form DNA. So, as you can see, the conditions required to produce the A form DNA is that it has 75% of water content, whereas if you increase the water content, it gives rise to a B DNA or a 92% water content. Franklin produced X-ray diffraction data of this B form of DNA, whereas Wilkins produced the data for the A form of DNA. So, the only difference is majorly the water content, and because of this water content, certain parameters of the DNA changes. The helix direction is right-handed. The average bases per turn, however, in A DNA is 11, so it is more compact, but in B DNA it is 10, it is a little stretched. The rotation per base pair is 32.7 degrees in A DNA, whereas there in B DNA it is the 36 degrees. The distance between the adjacent bases is 0.26 nanometers in case of A DNA and 0.34 nanometers in B DNA. So, the Watson and Crick model of DNA which we were looking at was basically the B DNA structure. The diameter of the DNA is 2.3, so A DNA is wider and a little compact, whereas B DNA has approximately 2 nanometers of diameter. The overall shape of A DNA is short and wide, whereas for B DNA is long and narrow. Another form of DNA is found inside the cells, which is called as the Z DNA or Z DNA, the alternating, where they have an alternating purine and pyrimidine bases. It has a left-handed helix and the average bases per turn is 12. So, it is quite longer structure. The rotation per base pair is minus 30 degrees, obviously because it is a left-handed helical structure, so it is in minus. The distance between the adjacent bases is 0.37 nanometers and the diameter or the width of the DNA molecule is 1.8 nanometers. So, it is again a most elongated one, but the basic difference is that it is a left-handed helical structure and the overall shape is basically elongated and narrow one. So, this is about the multiple conformations of DNA. So, in today's lecture what we talked about, what we looked at was that what were the experimental evidences that stated DNA, that assigned DNA its most important role, which is the genetic information, the heredity passing on from one generation to another. We looked at how the DNA structure was discovered, how the Watson and Crick famous model of DNA came into being and how different scientists contributed for this final structure of DNA, and we also looked at certain physical and chemical properties of the DNA and finally the multiple conformations of DNA. So, we will continue with this in my next lecture. I will continue by stating that how now the information is encoded inside the DNA. So, we say that DNA carries the information from one generation to another, but how is it coded? How does it contain that information? And then how is it transmitted from one generation? What are the mediator molecules which are responsible for transferring this information from one generation to another or from parent to the offspring? So, we will continue with this in our next lecture.
I would like to thank Dr. Charu Dogra Rawat for a very enlightening lecture. This lecture answers several questions regarding the discovery of DNA, regarding the structure and how regulation occurs in DNA, but it also ignites several questions in my mind and of course in the minds of our viewers. So, for one question I would really like to ask you is that DNA has a very simple vocabulary, or at least just 4 words. How does it give rise to the complexity in the regulation and in the expression?
See, when the tetranucleotide hypothesis, which was proposed by Levine and the earlier scientists actually believed that it contains just 4 bases A, G, C and T. So, how can that give rise to the variations? But now we know that these variations arise because of the arrangement of these sequences. So, a particular gene is composed of many number of these nucleotides which can be arranged in a different order to give rise to such kind of structures which is there. Moreover, on the top of it, not just the sequence but the structure of DNA, as I was talking about, that it will lead, it's not like a double helical structure which is present inside the nucleus. It has a higher order structure and that structure itself give a kind of a regulation which is called as the epigenetic regulation, that how the genes are expressed. So, a variety of proteins can be produced and therefore that gives the complexity to an organism. So
I'm sure in the coming lectures we'll be looking at the DNA complexities and how it gets its condensed form with the discussions with Dr. Dogra. Another question I would like to ask would be regarding how do you, when you showed the X-ray crystallographic structures, pictures of DNA, we could see that A DNA was very different from B DNA. So the bands that you suggested, does that suggest the stacking, extent of stacking, which is more present in A as it was stacked, and in B being elongated, we missed or we had loss of stacking?
See, that's what I explained that's why I included the multiple conformers of DNA here, because when you crystallize the DNA, there can be a variation in the water content, and therefore you can either have an A form or a B form DNA. So as already shown in the characteristics of A form DNA, in the A form DNA it's more compact. So when you do the X-ray diffraction pattern, it has the possibility basically for more stacks to appear in that particular area rather than in B DNA, because in B DNA the stacks are widely distributed. So when the stacks are a little widely distributed, for every turn there are 10 bases which are present, for example. In case of A DNA, it's more compact. So then you will have more number of stacks in a particular area than what they will meet inside when they touch when the X-ray is bombarded on the B form of DNA. So the difference in the pattern was that, but although both the structures very clearly showed that it is a double helical structure, it's a very consistent structure, and that the bases are stacked one over the another, only the differences between the physical features, the parameters, that probably there are more number of bases and there are less number of bases which are there.
Here I would like to ask another question, which is of course very sci-fi to me. Like when you were suggesting that DNA being chopped up was taken up by the bacteria, does this kind of transformation occurs in higher organisms also?
See, in fact in DNA, in the taken up by bacteria also, it's not like that, you add the chopped DNA inside the bacteria and it will take up. The bacteria has to be made competent. The bacteria are usually in their normal environment, this competence is very low, although it is present. And in the same manner, we can make the higher cells, and the higher cells are also made competent by certain chemical treatments or physical treatments by which they can take up exogenous DNA which is present inside in the surroundings. So naturally, it's a very, very slow phenomena. It's not a very fast phenomena, but artificially if we make them competent enough, they can take exogenous DNA, and it does happen in the eukaryotic cells also.
Ma'am, may I ask another question?
Yeah, sure.
When we were discussing the experiment where we had boiled off the DNA and it still retained its function, when we look at various biomolecules, most of them seem to lose their function upon heat treatments. So what is it in the DNA that makes it so survivable?
Unnati, that's a very interesting question, because that's actually intrinsic, it's in its structure. The DNA is, as I told you, has two chains which are wind together on each other, and we know that A pairs with T and G pairs with C. So an important aspect of this helical structure is the complementarity. And complementarity is the one that when you heat boil a DNA, then it denatures. That denatures basically means the melting. The two strands will separate, but DNA has this physical property that when you cool it, or when you bring down the temperature, the DNA, because of its complementarity of base pairs on the two strands, just zips up again. So if you do that slowly, then the renaturation is also achieved. So when they heat boiled the bacteria, where they heat boiled the DNA got fragmented. The bacteria become avirulent, but the DNA itself couldn't be damaged so much, because when you cooled it down and introduced it into another bacteria, it took its original form. But that has to be done very slowly, and that is basically for the smaller DNA. If you have a very large DNA, then there will be portions which will remelt appropriately, but there will be portions which will not be. So in this case, because it was chopped off, there were small fragments, and therefore they could be really renatured very quickly.
Thank you, Dr. Dogra. With this discussion and the lecture before it, we came to know several aspects about the DNA structure, and also we are now touching on its function. So with the subsequent lectures coming, we hope that many of our doubts, many of our questions will be answered with discussions with Dr. Dogra. And as in my mind, surely you must also be having many questions coming up to your mind. Would you like to have a discussion or answer which Dr. Dogra would like to show light upon? Then definitely do write to us on the email addresses displayed on the screen. Thank you viewers for joining us for the EduSat lecture.